just stalling a slight bit to make sure that we have a chance for people to come in, but we'll get started. I just wanted to lead off by thanking all of you for joining us today, both in person and on Zoom. Delighted to welcome everyone to the 7th Annual Barondes Lectureship. So this lectureship and celebration was established in 2017 to recognize and celebrate Sam Barondes, his remarkable contributions to the department, to UCSF, to the field of psychiatry, and to the neurosciences broadly. Sam has been a true pioneer from the very start of his career. His early work in the laboratory really laid the foundation for the pursuit of molecular mechanisms underlying serious mental illness. As chair of UCSF and chair of psychiatry at UCSF starting in 1986, has set his sights on identifying and recruiting the very best young physician scientists in the world, doubling down on the bet that cellular, molecular, and developmental neuroscience and human genetics would provide key insights into the mechanisms of psychiatric disorders and an avenue to developing better, more rational, more effective treatments, and truly to transform our field of psychiatry. It is remarkable every year that I give this lecture how prescient this vision was. And while the road, I think we would all acknowledge, has been a long and winding one, and no doubt longer than Sam had hoped when he started out, we are now, I think, at truly an inflection point, beginning to finally realize the promise that he recognized so early. Along those lines, I can't think of anyone who has made a more seminal contribution to our field or played a more central role in realizing Sam's vision than our speaker today, Eric Nestler. Eric received his BA, PhD, MD, and psychiatry residency training, all from Yale University. He served on the faculty at Yale from 1987 through 2000. He moved to Dallas in 2000, where he was the Lou and Ellen McGinley Distinguished Professor and Chair of the Department of Psychiatry at UT Southwest. He stayed there until moving back to New York in 2008. Throughout his career, his research has focused on the molecular mechanisms of drug addiction and mood disorders, and by every measure, his contributions have been literally off scale. He's been cited about 190,000 times. He's authored more than 700 publications, and he has an H index of 217. I didn't know it went that high. He's won countless awards and is an elected member of both the National Academies of Medicine and the National Academy of Science. He's also a fellow of the American Academy of Arts and Sciences. What is equally remarkable has been his contributions as an institutional and academic leader. At Yale, he was the founding director of the Division of Molecular Psychiatry. After chairing the Department of Psychiatry at UT Southwest and moving to Mount Sinai, he was chair of the Department of Neuroscience, director of the Friedman Brain Institute, and dean for academic and scientific affairs. He's the past president of the American College of Neuropsychopharmacology and the Society for Neuroscience. He has recently now been named the Anne and Joel Ehrenkrantz Dean of the Icahn School of Medicine in Mount Sinai, I think warranting a cognitive exam potentially, where he serves as Chief Scientific Officer and Executive Vice President of the health system. Again, I can't think of any psychiatrist over the last 2 decades who has simultaneously had more of an impact both in the laboratory and as an academic leader in our field. We're thrilled, as I know Sam is, that you were able to join us today to talk with us about your work on transcriptional and epigenetic mechanisms of drug addiction. Eric, thank you so much for being here.
Thanks, Matt. Thanks very much, Matt. Gosh, after that introduction, I think I'm done, right? Just stop. It is a tremendous pleasure to be here today, and I wanted to start by sharing this picture, which I think Sam was in Aspen maybe 15, 20 years ago. But just to give me the opportunity to acknowledge Sam as a longtime friend, colleague, and mentor. Sam and I met, we were reminiscing, I think 40 years ago at literally the 2nd Avenue Deli on the Upper East Side of Manhattan, where Paul Greengard, my PhD mentor, had joined us as well. Sam had just become chair at UCSF. I had just become an assistant professor in psychiatry at Yale, and Paul wanted us to meet. And Sam has been supportive of me ever since, every step through my career. So this is really special, Sam, for me to be able to give the Sam Barondes lecture. Congratulations. I also wanted to congratulate you all on this spectacular building and the Department of Psychiatry, Sam's legacy, Matt's current vision, really just outstanding sense of achievement and promise in the field of psychiatry. So it's just a great pleasure to be here. What I'll do today is give you a bit of a historical perspective on some of the work we've done looking for the biological basis of drug addiction, and then tell you about some more recent work. So years ago, we and other people proposed that addiction could be viewed as a form of drug-induced neuroplasticity based on the stability of the behavioral abnormalities that we know characterize an addicted state. We all proposed that an important role for gene expression in that process. So according to that view, a drug of abuse binds to its initial protein target, which are all at the extracellular aspect of synapses. And those actions lead to the initial effects of drugs, but repeated drug exposure required for an addiction begin to induce plastic changes inside of nerve cells that signal eventually to the cell nucleus. I have my finger on the keypad for the arrow, which I see perfectly on the screen. So we don't need, well, that's okay, I'll just use this. Making me look bad, thank you. Thanks a lot. So with repeated drug exposure, the drug effects eventually signal to the nucleus, where over time, gradually, progressively, they begin to produce changes through actions on transcription factors, proteins that bind to DNA in a sequence-specific manner, the involvement of many other nuclear proteins, as we'll discuss, leading to stable changes in the expression of target genes that somehow underlie the very stable changes in neural function that underlie the behavioral abnormalities that characterize addiction. It's an important point that all of the available treatments and most of the experimental therapies that are being examined for the treatment of addiction focus on extracellular proteins, despite the literally thousands of proteins that are post-receptor that could also be involved. And I'll come back to that at the very end of my talk. In order to understand the transcriptional mechanisms of addiction, one needs to know what parts of the brain to examine, and for that we've relied on a very large literature, this is familiar to many of you, of so-called brain reward regions, sites in the brain that evolved to mediate individuals' responses to natural rewards in the environment that are somehow commandeered, taken over by repeated drug exposure. This is a sagittal section of a rodent brain, but a human brain is highly homologous, focusing on dopaminergic neurons in the ventral tegmental area of the midbrain and the projections of those cells to the limbic forebrain, the field focusing, as I will today, on the nucleus accumbens, but also highlighting the critical involvement of many other dopamine target brain areas, such as those shown in this slide. And historically, my lab focused on two transcription factors. One is delta phosphB, which we came across by looking at the ability of drug exposure to induce in brain immediate early gene transcription factors like Phos and related proteins. We found, going back several decades now, that in response to a single drug exposure, there's a very rapid induction of Phos and these related proteins. With delta phosphB, a modified isoform of the phosphB gene product, it induced only to a small extent in response to initial drug exposure. But unlike all other Phos family proteins, which are highly unstable, delta phosphB is highly stable, and that imbues it with the ability to accumulate in response to a chronic drug exposure. So with repeated drug exposure, you see a stepwise increase in levels of this protein, and we showed that delta phosphB actually feeds back and suppresses the expression of these acute Phos proteins, leading to what we called at the time a molecular switch. CREB is the other transcription factor that we identified historically, based on work that had shown that changes in the cyclic AMP pathway mediate the effects of drugs of abuse on cultured cells first and later in the brain. CREB being one of the transcriptional output pathways, effectors for the cyclic-AMP pathway. And we found that CREB's temporal properties are much more typical in showing a spike and trough pattern in response to acute and then chronic drug exposure. And so the first 10 or 20 years of my lab basically focused on trying to decipher what function delta phos-B and CREB served when they were induced or activated in the nucleus accumbens. And this slide summarizes those data. And I want to give a shout out to two early postdocs in the lab, Bill Carlazon, who was recently president of the ACMP. That was a nice thing to note. And then David Self, a longtime postdoc and then faculty colleague of mine in Dallas. And what we and Bill and David and many other people found is that delta phos-B is induced selectively in one subtype of nucleus accumbens neuron called the D1 type medium spiny neuron. I'll talk more about that in just a few minutes to define that. And we showed, using a variety of genetic and viral tools, that induction of delta phos-B in the D1 subset of medium spiny neurons increases in animal sensitivity to drug and natural rewards, creates a positive emotional state, and drives drug craving and relapse, presumably through a process of positive reinforcement. In many ways, CREB is the inverse. CREB is a classic mediator of negative feedback homeostasis, contributing to drug tolerance and dependence. CREB seems to be induced equally in both D1 and D2, the other subtype of medium spiny neuron in the nucleus accumbens. Once induced, it has the opposite effect. It decreases in animal sensitivity to drug and natural rewards, and also drives drug craving and relapse, although through a process of negative reinforcement. Beginning about 20 years ago, we wanted to move beyond transcription per se and look at the state of chromatin and how transcription factors mediate changes in gene expression through what are called epigenetic mechanisms. That's depicted here, and again, this is by way of review for many of you, but I'll go through it just very quickly. We know that a mammalian genome has about 3 billion nucleotides if stretched out linearly. That's about 2 meters in length. Somehow that 2 meters of DNA is compacted within a microscopic cell nucleus, really through a triumph of evolution. And the field of epigenetics has partly solved the mechanisms through which that compaction occurs. That is shown in this slide, where the DNA double helix is wrapped around octamers of histone proteins to form the unit of chromatin called a nucleosome, which is then further organized in very rote ways in the most compacted form, creating what we would recognize as a chromosome. This is very useful from an experimental point of view because DNA can only serve as a template for RNA transcription in where it is relatively open in the context of nucleosome structure, whereas in more closed structures, the DNA is functionally inactive. And that means that we can use epigenetics really for the first time to gain insight into transcriptional mechanisms in the brain in vivo, where prior to this point, any study of transcriptional mechanisms required work in cultured cells. So our proposal at the time was that drug addiction, other types of behavioral change, could be seen in some way by some analogy to developmental biology and cancer, where a stimulus would create a stable change in epigenetic structure, particular segments in the genome in a given cell type, leading to very long-lasting changes in gene expression, driving the stable changes in behavior that I've outlined before. And so giving a little more detail to that mechanism, this is showing this level of analysis. Now looking at the nucleosomes, or these spheres with the DNA wrapped around them in a relatively open state versus a relatively condensed or closed inactive state, showing the actions of transcription factors like delta phos-B and CREB binding to relatively open areas to their response elements, then recruiting to the genome literally hundreds of proteins that mediate the activation of a gene, or in concert also recruiting several hundred proteins to repress chromatin at distinct sites of the genome. So the approach that my lab started using over the years was to overlay different platforms of next generation sequencing to identify these stable drug-induced changes in gene expression and the underlying epigenetic mechanisms involved, creating what we would refer to almost like a template periodic table of the entire landscape of gene expression modifications that might be occurring in a given tissue. So according to that idea, we would use RNA sequencing, say focusing on the nucleus accumbens or any other region, to look at sets of RNAs that are induced, another set that are repressed in response to cocaine exposure. If an RNA is induced through transcriptional mechanisms, it will be associated with some epigenetic modification associated with gene activation. That might mean an acetylated histone or some other type of chromatin modification. And overlaying on top of that, transcription factor binding with the transcription factors that were implicated in drug action. This is an enormous bioinformatic challenge. Literally 20 terabytes of data would be represented in the cartoon shown in this fashion. And to be honest with you, 10 years later, we're still working toward the tools that are needed to optimally derive information from such complex data sets. But I think that they've taught us something fundamentally new about the addiction process, importantly, allowing biology to guide our studies as opposed to guessing what might be the more important mechanism to study. I'll make that point in just a minute. So let me just tell you about an experiment we did a number of years ago. This was done by Dina Walker, who's at Oregon, and Erin Kalapari, who's at Vanderbilt. And what they and others did was to take a group of mice, have them self-administer cocaine or saline for a period of time. We harvested brain areas 24 hours after the last self-administration experience. Animals were returned to their home cage for withdrawal. And then a month later, the animals were given a single challenge dose of cocaine or saline and analyzed 1 hour later. We analyzed 6 brain areas, the same brain areas that I've already outlined in a prior slide. This 30-day withdrawal time point is important because in the field of addiction, both in animals and in humans, it's been shown that the degree of craving in a human, or the degree to which one can demonstrate an animal is seeking, wants, works for cocaine, increases during that withdrawal phase. That's shown in the bar graph to the right. So if you take an animal 24 hours after self-administration, put them back into the self-administration chamber, now the cocaine lever is turned off, but it's otherwise the same context. The animal will press the lever previously paired with cocaine. The interpretation is the animal is trying to get cocaine. It's seeking cocaine. And you can see that response is dramatically increased 30 days later. The people in the field call that incubation of cocaine craving. So we're particularly interested in what are the time-dependent changes that are occurring as a consequence of long-term withdrawal that drive the sustained, maybe even heightened state of craving. This is just showing you one example of the type of data that we would obtain. This is looking at bulk RNA sequencing of nucleus accumbens tissue from animals at these various time points. So this is 24 hours after the last cocaine self-administration. This is 30 days after saline self-administration, followed by an acute dose of cocaine, analyzed 1 hour later, essentially an animal's first ever exposure to cocaine, with animals analyzed an hour afterward. Chronic cocaine self-administration, 30 days withdrawal, followed by a saline dose, but placed back in the self-administration chambers. And now an animal 30 days after chronic cocaine self-administration, put back into the chamber with a challenge dose of cocaine. And then using a machine learning to identify sets of genes that are uniquely regulated after 30 days of withdrawal, whether an animal received a saline or cocaine challenge dose. Or to the right, you see in particular what's unique about an animal's transcriptional response in nucleus accumbens to this challenge dose of cocaine. Let me point out one thing about this slide. The second heat map here and the bottom heat map here represent an animal 1 hour after a cocaine challenge dose. The only difference between the two conditions is the bottom heat map are animals that had a distant history 1 month prior of chronic cocaine self-administration. So you could see the rather dramatic effect of that distant history of cocaine self-administration on the transcriptional response of nucleus accumbens. And so an interest in the lab is to understand what is driving this enhanced primed expression or the desensitized expression under these conditions. Now we can go one step further. Even though these experiments were done on genetically identical C57 animals, there is broad behavioral variability in their features of self-administration. So this heat map shown is looking at different behavioral degrees to which an animal pressed a lever, active lever versus inactive lever, and a whole range of other details of the self-administration process, using, again, machine learning to create what we call an addiction index. Now, addiction is a big word for a mouse. Really have no idea if a mouse gets addicted, but it's looking at addiction-like behaviors as best you can in a laboratory animal. And what you see is that animals that self-administered saline all had a low addiction index. Animals that self-administered cocaine had quite a variable response. Some showing quite high levels of that index, others lower, even though every animal achieved what would be considered in the field replicable, significant self-administration of cocaine. So then we related the gene transcription data with the behavioral data, and I'm showing you that relationship here. So now we're looking at genes whose expression levels are correlated in the different experimental conditions that I've already analyzed, that where the regulation is correlated positively with the addiction index under the red bar, or correlated negatively under the gray bar. And in the nucleus accumbens in particular, an interesting pattern becomes apparent. Notice how the animals that received their first ever exposure to cocaine, that's this heat map on top, compared to the heat map of animals also responding to an acute cocaine dose, but with a history of chronic cocaine self-administration, opposite regulation of most of the same genes, which is interesting because what that means, what that suggests, is that one can determine the addictiveness of cocaine in an individual based on the individual's transcriptional response in the nucleus accumbens to that first ever cocaine exposure, even if the degree and sign of changes might be opposite in the chronic state. We were also able to interrogate this data set and ask what are the predicted transcription factors upstream of the genes indicated in this heat map. This shows the list of the top ranked genes. We were delighted to see highly ranked regulation of CREB, AP1 family, again, that's delta phos-B in the chronic cocaine condition. But we also identified many additional transcription factors that are highly implicated, and the ones in red are the ones that we had more recently gone back and validated this bioinformatic deduction with experimental data. Now, everything at this point was done on bulk RNA sequencing, even though we know that the nucleus accumbens is a cellular heterogeneous tissue. At the time, it was really not feasible to look at individual cell types. But more recently, we've taken advantage of this cellular heterogeneity. So let me tell you just a little bit more about the D1 and D2 medium spiny neurons. In general, a large literature, and I'm just highlighting a few papers from my lab here, but many investigators have contributed to this work. I want to acknowledge Mary Kay Lobo, who's at Maryland, Erin Kalapari, again at Vanderbilt, and then Bowen Tan in Jeff Friedman's lab at Rockefeller, and Caleb Brown in my lab. Bowen is now at the Allen Institute, and Caleb is now at the University of Toronto. What the field has shown largely is that using optogenetic or chemogenetic tools Activation of D1 medium spiny neurons generally promotes rewarding responses to cocaine or other drugs of abuse, while activation of D2 cells in general has the opposite effect. And this has been done pretty well established across many experimental paradigms. Using more recent tools like fiber photometry, it's made it possible for the first time to record the activity of specific types of neurons from awake behaving animals. It's also been able to show that in general, cocaine has opposite effects on the activity of these nerve cells in the intact brain. In general, acute and chronic drug exposure tends to activate D1 medium spiny neurons and tends to do the opposite to D2 medium spiny neurons, although the reality is much more complicated than this short-term version I just gave you. Finally, I want to highlight that the D1 and D2 medium spiny neurons defined by the dopamine receptor subtype enriched in these cell types mostly, not perfectly, mostly corresponds to the direct and indirect pathways identified anatomically. So the D1 cells tend to overlap with the direct projection pathway, D2 neurons, the indirect projection pathway. More recently then, Philip Muse in the lab, and Philip is now on faculty at Boston University, we have essentially repeated the types of experiments that I've described to you already, but selectively on sorted D1 and D2 medium spiny neurons. And some of the early data are shown here. What Philip did first was to use a method called ATAC sequencing. ATAC sequencing is simply a technology that allows one to determine the degree to which nucleosomes are opened or closed. Higher amplitude means more open, comparing the same conditions of cocaine exposure that I've already described to you. First, comparing D1 and D2 cells, you can see that at baseline, the pink line, D2 medium spiny neurons have a more open chromatin on average. This corresponds with RNA sequencing data that shows a significantly larger number of RNA transcripts expressed by D2 neurons. Interesting. But notice how cocaine produces dramatically different effects in the two cell types. In response to acute cocaine, there's a dramatic increase in the opening of chromatin, average genome-wide in D1 cells, not seen in D2 cells. That is sustained during 30 days of withdrawal and opens still further after that priming dose of cocaine. Now, these are genome average data, which means I could go in and identify a gene in both of these cell types that might show robust regulation, but this is genome-wide. Yeah. I'm sorry, Dort?
Did anyone, can you hear me?
Yes.
Did you or anyone else do the same type of experiment or similar, but in the DMS or DLS?
Oh, you mean in the striatum, in the dorsal striatum?
Because they also contribute to addiction.
To a point. Other groups have mostly looked at the dorsal, medial, and lateral striatum. And cocaine produces dramatic effects in those regions as well. In fact, in the earlier study, the dorsal striatum was one of the tissues we looked at. One sees interesting effects there too. I'm sorry? There hasn't been the same careful characterization of D1 versus D2 medium spiny neurons in dorsal striatum. We then complemented ATAC-seq with RNA sequencing, that's shown in this slide, and one can demonstrate dramatic gene priming in D1 medium spiny neurons, not nearly as apparent in the D2 medium spiny neurons. So cocaine's transcriptional effects really dominate in the D1 medium spiny neuron subset. And one of the really interesting and gratifying findings of this data set was that the third topmost prime gene observed in D1 medium spiny neurons was the FOSB gene. So again, even though we studied FOSB decades earlier through a hunch, sometimes hunches prove to be correct, but this is so much more convincing because it's following the data sets and allowing biology to guide us on what's most important. The next question becomes, what is it that is working in concert with the transcription factors that I've been talking about that drive this dramatic gene priming in D1 medium spiny neurons? So if we rewind about 15 years, we started doing chip sequencing, a method that enables one to characterize chromatin modifications genome-wide on the types of histone modifications, different types of histone acetylation, methylation, and so on, that ENCODE and other data sets had demonstrated being the most important epigenetic mechanisms, mostly in peripheral tissues, cultured cells, cancer cells, and some peripheral tissues. And we published lots of papers with significant findings, but they were all unsatisfying to me because the degree of overlap between any given histone modification, for example, with an RNA sequencing data set was less than 10%. So there was a contribution of the highly studied chromatin marks, but not a dominant effect. So I give credit to Phil Muse, the postdoc at the time in my lab, who collaborated with Simone Sidoli at Albert Einstein and to interrogate to take an open-ended approach, to not guess what chromatin modification might be the most important, but to use a proteomic approach and identify which histone modifications are most dramatic. A small segment of that data, because there are hundreds of different histone modifications and combinations of histone modifications, so a small subset of those data are shown in this slide. And by far, the most dramatic change that we saw, again, under the three major cocaine treatment conditions that we've studied, an acute cocaine exposure, long-term withdrawal after chronic cocaine, an acute cocaine challenge in the context of distant chronic cocaine exposure, was regulation of a histone variant protein called H2AZ. H2AZ had never before been studied in addiction. One group, Eva Zavchik in Toronto, has studied it in hippocampus in the context of learning and memory, and that's it for its literature in brain. So we were fascinated by the fact that H2AZ was dramatically induced in nucleus accumbens in response to acute cocaine and broadly depleted in this 30-day withdrawal condition. We coined the term chromatin scars to indicate the specific change in a histone modification or other chromatin modification that persists for a long time in response to an environmental stimulus, H2AZ depletion representing sort of an example of that chromatin scar. So the first thing Philip did was to perform chip sequencing on nucleus accumbens tissue for H2AZ and its acetylated variant, H2AZ acetylation. Under the same four treatment conditions that I've already described, you can see that under control conditions, there's some baseline level of H2AZ incorporated into nucleosomes. I should say that H2AZ is a variant substitute subunit of the H2A subunit that's part of every nucleosome. So every nucleosome in the genome has two H2A subunits, and then H2AZ is substituted in some regulated process that remains only partly understood. In response to acute cocaine exposure, there's an induction of H2AZ, but under these two chronic conditions, there is broad depletion of H2AZ. This is the kind of result that I'd like to see. I mean, this is the most dramatic effect that we've seen in the lab over 30 years, I would say. And now when we overlay the chip sequencing data set with our RNA sequencing data set, now we see broad 50, 60, 70% overlap between genes that show H2AZ depletion and genes that show priming in response to a challenged dose of cocaine with a history of prior chronic cocaine exposure. We took advantage of the fact that there is a protein in the genome called ANP32E that's a chaperone, one of just a couple chaperone proteins that removes H2AZ from nucleosomes, never before studied in brain, but characterized in peripheral cells. We interrogated our data sets that we had available and found that, in fact, the AMP32E, the chaperone protein that removes H2AZ from nucleosomes, is induced in D1 medium spiny neurons selectively in the chronic cocaine condition. And we took this as strong evidence that perhaps AMP32E induction is mediating the depletion of H2AZ that we had identified, and we obtained the data on the right supporting that hypothesis. This is just a simple Western blot experiment on chromatin tissue isolated from nucleus accumbens, showing that after chronic cocaine exposure, there's the loss of H2AZ from the chromatin fraction. Again, it's not a subtle change. You see it on Western blot, and you can completely block that change when you knock down AMP32E in nucleus accumbens neurons using a viral vector approach. Having that validation in hand, we then used the viral vector knockdown to interrogate what role this H2AZ depletion might be playing in the behavioral and transcriptional actions of cocaine. So first, the behavioral effects. Using a condition-place preference paradigm where animals learn to prefer an environment paired with cocaine, one can see that under control conditions and a relatively high dose of cocaine, if you look at the open bars, there's a strong increase in the degree to which an animal stays in the cocaine-paired environment. When one knocks down AMP32E in D1 medium spiny neurons, you broadly inhibit the rewarding effects of cocaine. One can demonstrate that in a self-administration assay as well. Animals self-administering cocaine stably, now making it more difficult to get a dose of cocaine. Instead of pressing the lever once for each dose, having to press the lever twice for each dose. Normal animals will double their response and press the lever twice as often, a response completely blocked by AMP32E knockdown. This is selective for D1 medium spiny neurons, as we would expect, because when we knock down AMP32E, we prevent any depletion of H2AZ in D2 medium spiny neurons. We do not see a reduction in cocaine reward. In fact, we might even see the opposite effect. One can demonstrate the role of H2AZ depletion in the transcriptional response to cocaine as well, doing RNA sequencing on the tissue of nucleus accumbens from the animals that I had just described to you. We can show that in this chronic cocaine condition with a cocaine challenge, one sees the induction of genes or the repression of genes as one would normally expect. And when we prevent the H2AZ depletion, you broadly block that type of priming and desensitization. So again, a dramatic effect implicating H2AZ dynamics in the long-lasting transcriptional and behavioral effects of the drug. So our model then is depicted in this slide, that under control conditions, as in most genomes, there's ambient levels of H2AZ incorporated at particular sites across the genome in a cell type specific manner. The first ever exposure to cocaine acetylates H2AZ, seems to contribute perhaps to the acute effects of cocaine on gene expression. But after long-term cocaine exposure and long-term withdrawal, there's the broad removal of H2AZ from the genome. We think that's mediated through the induction of AMP32E, this chaperone protein, leading to a prime state so that in response to the next cocaine exposure, there's a much more dramatic transcriptional response. In the last few minutes of my talk, what I'd like to do now is to take you through a few more recent studies that we've done, trying to further explicate the complexity of nucleus accumbens medium spiny neurons beyond the D1, D2 dichotomy, which we know exists from several laboratories. So the first approach to do that was a study published recently by Marine Salary, who's now in Marseille, using single-cell sequencing from mice in which the ARC promoter, ARC is immediate early gene promoter driving Cre recombinase. It's a estrogen receptor dependent Cre recombinase. Other labs have done similar experiments using other immediate early genes driving Cre, for example, FOS-Cre, even MPAS4 Cre. These three immediate early genes are interesting because they do seem to reflect the activation of the same cells, but a broader and broader subtype. So ARC shows the broadest induction, then FOS, then MPAS4 much smaller. But broadly speaking, in response to an acute dose of cocaine, one sees ARC Cre activation way less than 10% of D1 medium spiny neurons. It's a sparse response, very unsatisfying personally, but it's a sparse response. So one hypothesis in the field was, well, these sparse neurons are still behaviorally very important. I think that's true, but I also think that focusing with the immediate early gene promoter is missing a lot of other activated cells, and I'll come back to that later. So using single cell sequencing of the entire tissue, you see the range of cells that you'd expect to see. When we do single cell sequencing on the ARC Cre positive ensemble of cells induced by an acute cocaine exposure, one sees this picture of cellular heterogeneity, not solely D1 cells as we would have predicted. About 75% D1 cells, about 25-30% D2 cells. So there's a subset of D2 medium spiny neurons that had gone unnoticed by us using the other methods that I've depicted. Interestingly, the immediate early gene promoter driven cells, the ensemble decreases in size with chronic cocaine. Again, people would have predicted the ensemble would increase with chronic cocaine. It's the opposite. It gets a smaller and smaller ensemble with mostly different cells involved in the acute or chronic state. Interestingly, regardless of whether the cells are D1 or D2 type, they all show an enhanced excitability after chronic cocaine exposure. That coincides with the fact that we can identify, as shown in the overlap of this Venn diagram, a transcriptional pattern that intersects the D1 and D2 cells that are part of the ensemble. So the ensemble cells have a transcriptional pattern distinct from that of all the D1 cells or all the D2 cells, just adding a complication to the prior analyses. And then finally, we were able to determine what functional role these immediate early gene ensembles play in cocaine's behavioral responses. Using correlative experiments, one can show that the size of the acute ensemble correlates positively with condition-place preference responses. The bigger that acute ensemble, the more rewarding animals find cocaine over a period of time. But you see the chronic ensemble is even the opposite. The chronic ensemble really seems to play an adaptive homeostatic response in opposing cocaine's actions. One can demonstrate causality using optogenetics. But as I said, this is rather unsatisfying because it's a sparse number of cells. So what Philip Muse did next was to simply fact sort all D1 cells and all D2 cells, did single cell sequencing on these pooled samples of D1 or pooled samples of D2 cells and identified subclusters of D1 cells, subclusters of D2 cells that are showing consistent transcriptional patterns across the same 4 chronic cocaine treatment conditions that I've described to you before: control, acute, chronic, chronic plus acute. I'm showing you here just the transcriptional pattern of genes activated or reduced in D1 in one cluster called cluster number 1 in the chronic plus acute paradigm. But you see very different responses in the other clusters. So this D1 cluster is interesting because this dominates in animals that are in the chronic withdrawal condition. So it's a cluster that is identifying D1 cells uniquely regulated transcriptionally in that chronic withdrawal state without a prior cocaine challenge. This cluster number 7, numbered randomly, is an interesting cluster because it concentrates on cells that are activated at a time of an acute cocaine exposure, whether it's a single acute exposure or chronic plus acute exposure. In fact, this is where the immediate early genes sit. So this cluster here, D17, are the ARC-3 cells that I've already told you about. All the other clusters are just different ensembles that are invisible in the brain with the other methods. And here's an example of a D2 cluster, D2 medium spiny neuron cluster that is particularly activated in this chronic acute condition that remained invisible to us as well. Now, the real challenge with this experiment is there is no way technically now today for us to go into these cells and capture the optogenetic activity or the fibrophotometry activity of these various clusters. So technical innovations are going to be required for us to draw those causal connections. And then finally, using a tool that many of you use particularly effectively here at UCSF is single-cell calcium imaging of nucleus accumbens cells, focusing on recording from D1 cells or recording from D2 cells, just showing you a small smattering of the data. This is work done by Bowen Tan and Caleb Brown that I mentioned earlier. One can show that a broad effects of cocaine in activating cells, but in this experiment, like 40% of D1 neurons show a detectable calcium signal, far greater than what was apparent with immediate early genes. So the immediate early genes are just missing a lot of activated cells. Turns out that the cells activated by cocaine are the same that are activated by natural reward. So as the field had predicted, there is no specificity applied to nucleus accumbens neurons with respect to the type of reward an individual is receiving. One can then compare cocaine to other drugs of abuse. For example, this study compared the responses to morphine, where we found a dominant effect of cocaine in enhancing excitability of D1 medium spiny neurons, no real effect on D2 medium spiny neurons. The size of the D1 cluster increases, in fact, over time with repeated drug exposure. That does not happen in the D2 cluster, whereas morphine and opioids are very different. Opioids seem to have effects on both cell types. This was really interesting to us because the point I forgot to mention earlier was that delta phos-B is induced by every drug of abuse uniquely in D1 nerve cells, except for opioids, which induce delta phos-B in both D1 and D2. So opioids are unique with respect to all other drugs of abuse, including alcohol, in their ability, I said that for DORT, so that we could, in terms of their more broad effects on nucleus accumbens cells. And then transcriptionally, a study published by Caleb Brown verifies this at a transcriptional level. Broad overlap of the transcriptional effects of chronic morphine and chronic cocaine on D1 medium spiny neurons, broadly different effects on D2 medium spiny neurons. One more data slide, because I think it's particularly important in today's world, where the utility, accuracy, and validity of animal models is being questioned. I think it's valid to ask that question: to what extent does a chronic cocaine self-administration in a mouse recapitulate features of the pathology in a person with cocaine use disorder? The field has been forced to answer that question based on comparing behavioral abnormalities. I think that's difficult, comparing a mouse to humans. But now we have all these transcriptional data sets, so we can answer the question, to what extent are gene expression changes in nucleus accumbens or any other brain area that are occurring in a mouse with chronic cocaine self-administration recapitulating the pathology that we see in a human who has cocaine use disorder? So collaborating with Deborah Mash and her colleagues then at the University of Miami, Philip Muse and Ashley Cunningham did an RNA sequencing study of human nucleus accumbens neurons, and what they found was broad convergence in genes that are upregulated in the upper right, down-regulated in the lower left, genes that are regulated in the human nucleus accumbens of people with cocaine use disorder versus control subjects, and mice that chronically self-administer cocaine. And that convergence is not seen if one uses the acute cocaine data set. So to me, this is empirical evidence demonstrating that the molecular pathology of a mouse in the nucleus accumbens is recapitulating broadly what is seen in a human with cocaine use disorder, meaning that we can infer some of the biology of what's happening in human substance use disorder patients from our animal research. Now, I realize that in today's lecture, I was talking about a syndrome like addiction, which we know is so affected by social, cultural, and psychological factors. And here I'm not even talking about the brain, but about nuclei and chromatin biology. But I feel that the kind of data sets that we've been providing represent a map that others can utilize to identify the most important targets for drug discovery efforts. So one can use these data sets to identify regions of the genome that are undergoing these types of dramatic chromatin scar-based changes in gene expression in receptors, transporters, ion channels, structural proteins, and so on for therapeutics. But I will also highlight the fact that in cancer biology, some of the most exciting new therapies that are being tested in the clinic today are drugs directed against epigenetic mechanisms, which means that it would be possible today to do a study on patients with a substance use disorder with a molecule, say, acting against an H2AZ chaperone, as just one example, as long as that molecule is safe enough to give to a patient with a substance use disorder. So we think that that's a very viable approach and something that we're interested in doing at Mount Sinai. So with that, let me conclude by saying I hope I've convinced you that cocaine dramatically alters the transcriptional organization of the nucleus accumbens in a very cell type specific manner. I focus today on cocaine and nucleus accumbens, where we know that many other brain areas need to be studied, many other drugs of abuse need to be studied, in particular opioids. But I did emphasize the power of what we've been able to learn with unbiased, open-ended investigations. You know, as I've been emphasizing, allowing biology to guide what you study in the lab, not just choosing one protein of interest. I introduced the concept of chromatin scars as mediating the long-lasting impact of cocaine on the genome, and also the idea that this provides a template for future drug discovery efforts. And I would expect us to be able to generate treatments that are both pan-addiction as well as drug-specific treatments, depending on a person's substance use disorder. So with that, let me stop. Thank you again so much for the opportunity to be here. Mark.
Eric, fantastic talk.
Thanks.
Really beautiful. I'm really struck by how the transcriptional effects that you're describing. It seems it's really a small fraction of the neurons in the tissue, right?
Mm-hmm.
So the calcium response is pretty broad, but the transcriptional response is pretty sparse.
Well, so I forgot to make that point. So let me just go back quickly. That the 40% of D1 medium spiny neurons that, for example, are represented here, we either can analyze these by looking at sorted D1 medium spiny neurons, as I showed in this slide, or by single cell sequencing of all cells. And what we can't do yet is to relate this 40% to transcription. Because as I mentioned in the text, but forgot to mention in my talk, there is no way that I know of, and maybe some of the aficionados here are developing ways, but there is no way for us now to capture cells that are showing a calcium imaging for then downstream transcriptional analysis. I know that efforts are underway to develop those tools, but to my knowledge, they're not yet available.
Okay.
But I think that answers your question.
Yes. Thank you.
Yeah, John, maybe Mark, just pass the microphone 2 rows up. Toss it, just throw it and see what happens.
The fascinating effect of this histone variant at the location of the regulatory elements where the action's happening. What do you think is so you say that the chaperone's taking that guy out of there. What is the biochemical effect, or even what is the attack-seek effect in terms of opening the chromatin in those locations?
So in other words, why is it that H2AZ is being depleted from a subset of sites, and not all sites?
What is the nature of the scar besides the depletion of the AZ?
It remains unknown. So we need to study that. It's not clear from the extant data in peripheral tissues that H2AZ depletion would be expected to enhance transcription, as we clearly observe and demonstrate causally. Both directions of change have been reported in peripheral tissues. So we need to understand now, and this is one of the ongoing experiments that we're doing, is to determine the factors that are recruited to the sites in the genome where H2AZ is depleted, and then what then drives transcription. There's broad overlap between sites of H2AZ depletion and delta phosB deposition. So we think that delta phosB is a pioneering transcription factor where H2AZ is being depleted at sites that concentrate around putative enhancer elements as outside of promoters. But what I just told you is really a work in progress and not ready to really present. Dorit, hi.
Hi, related to, I just remembered, because both of you asked questions, that the ride we took with you, Eric, with Mark, with John, in my little Mini. Remember that?
I did not hear that. I'm sorry.
We had a ride and we met Sam there in my little Mini. You were in the back.
That's right. I do remember that cab ride. It took like twice as long to get to the restaurant as expected.
Not the cab ride. I was the driver.
Oh, you were. That's why it took twice as long.
Anyways, related, I think, to Mark's question. You basically, cocaine changes the landscape of the genes, right? In D1 neurons, in this case. How do you get from there to, firing to behavior?
To destroying a life. Yeah.
I mean, it's...
No. So that is a great question. I focus today on the underlying transcriptional mechanisms, but another need in the field, something that my lab has also tried to focus on, are identifying these target genes that are mediating those effects. Ultimately, we really still lack the ability, I think, in neuroscience to understand exactly how it is that altered activity of a collection of nerve cells and activity of a circuit are driving a behavior. I think we're getting close. Some of the people in this audience are getting us closer to that. And it's an extremely exciting time for the field. That's where neuroscience needs to be today, in my opinion, for the next generation of researchers, to be able to draw causal relationships between the molecular constituents of a cell, their activity in a circuit, how a circuit drives behavior, how that circuit and behavior then alter the molecular constituents of the cell and feedback, and so on, right? But we, for the first time, have the tools to be able to complete that loop. It's an extremely exciting time. Yeah.
At a much more basic level, but
right.
At the same
As I said, in the old Star Trek, I'm a doctor, not a philosopher, but go ahead.
in these remarkable data sets that you have, and neurons that are changing their excitability, do you have a sense of the channelome changes that are happening within these neurons that might give you these long-term changes in?
Absolutely. So that's always one of the things that we look at in these data sets are the different families of channels and glutamate and GABA receptors, ionotropic receptors with ion channels that might be controlling the excitability of cells. We've even had the ability to go back and causally implicate some of those changes in the biology. I think the other challenge in the field is with these data sets, and by analogy, Matt, this may relate to some of the underlying genetic diatheses that we have. There are so many genes that seem to contribute to the risk and then the pathophysiology of these illnesses. And we need to develop tools that make it possible to interrogate the role of collections of many genes at one time and not go gene by gene, at least in a complex disorder like addiction, which may broadly lacks rare variants.
So, question here. Oh,
Raghu.
Oh, thank you.
Best friend of my daughter at Wesleyan, Connecticut, 10 years ago, 15 years ago.
I don't, yeah.
Yeah,
well,
who
looking
knows?
at
A whole
these
lifetime ago.
citations, it reminds me of when I was in the lab. So.
That's right. And he volunteered in my lab one summer too.
So actually, off of this slide, I'm curious if you could shed light on what is being done in the drug development and where we're at with that.
So there's broad interest in looking at many potential mechanisms for treating drug abuse. I think honestly, the field of psychiatry has too often taken a novel chemical entity and then tried it across syndromes. If it doesn't work in schizophrenia, let's see if it works in drug addiction. I hate to be so negative in that way, but I feel that we've lacked the precision of pathophysiology knowledge to make better guesses than that. On the other hand, it's the same in cancer biology, where a medication developed to treat, say, prostate cancer turns out to fail on prostate cancer, but it works very well in lung cancer, just as an example. So maybe we're not that far off from other fields. But I think where we're heading is to be able to finally have the pathophysiology to enable more specific possibilities to be developed and then derive the novel chemical entities required to study. The major limitation are the novel chemical entities. I give you a wealth of proteins here that one could study in a human with a substance use disorder, but we don't have the chemical tools to enable us to do that. So I think that is probably the greatest limitation in the field today. And then, of course, you read the Journal of the New York Times, and you know that GLP-1 agonists are going to treat drug addiction, which may be effective in treating some people with a substance use disorder with, at this point, almost no knowledge of the underlying biology. But that's okay, because often it's the human observation that then enables us to go back and look in an animal model to understand mechanisms.
Two questions.
We have to go with Dr. Barondes down here first.
This has been raised already, but it's daunting. The fact that there are so many different effects with so many different mechanisms, traceable to so many different pathways, it's daunting. I mean, I just don't see. You mentioned it's true also of polygenic disorders or multi-gene. It's daunting. You can hand wave and say, well, blah blah blah. Something will. I just don't see how you're going to get around it.
Yeah, I mean, it's definitely daunting. And this is just my lab. Now add to it 20 other labs that are creating similar parallel data sets that show still further complexity. And so putting it together is the challenge for the field, I think.
Yeah, and it's the same kind of problem as, so the other side is the clinicians. let's throw something at everything and see what happens. That's daunting too. And it really has not worked out very well.
Yeah.
To be honest, it's really not worked out very well. And I've watched it I keep thinking, I remember writing a book about, new ways to make, genetics and blah, blah, it'll all, it's really, I mean, it's nice to give dopamine for Parkinson's disease,
Yes.
but it's like,
It's true. I was telling some people earlier today that I've had a lab for almost 40 years. 40 years ago, Steve Hyman, Rob Malenka, and I, the three musketeers, boldly predicted that the tools of molecular cellular biology would solve all these problems within a decade, right? So here we are 40 years later, very humbly standing before you, not having done that. And, I think it speaks to, number one, the complexity of the brain and the complexity of the diseases of the brain. The experiments that we did 30, 40 years ago were really Mickey Mouse compared to what is possible today. I tell students at Mount Sinai that for my PhD thesis in Paul Greengard's lab, it was a pretty good thesis. The most advanced statistical test I used was a t-test. And I think it was valid because I had 10 rats, 5 control, 5 treated, measured 1 protein in the brain. That was it. This is a dumbass experiment, right? But all of a sudden now we've had these vast data sets. We're just beginning to learn how to use the data, where we really need computational experts to guide us on how to cross all 20 labs, all of our data sets, and really look at what's most important.
but if you look at it honestly, the fact that you have so many different mechanisms that you've identified, and plenty of others that you haven't identified, and they're all relevant. And your tool is, I'm going to very specifically craft it and kill that.
Right.
It's not going to do anything.
Right. It is one reason why I am enthusiastic about looking at transcriptional and epigenetic mechanisms of treatment, because at least when you affect a transcription factor or histone molecule, you're going to be affecting hundreds of proteins downstream eventually. So maybe that would be more effective, but that remains to be seen.
More side effects.
And more side effects too. Yeah. But one has to be optimistic.
We share that to our benefit and to our regret.
Eric, may I?
Yes, please go ahead. Go ahead, Alan. Go ahead.
Two quick ones. I'm very curious about the cycling of delta phos B.
Yeah.
There's the process, there's an addiction process. I thought we're not supposed to use the word addiction anymore and use misuse. That's an aside. But I'm curious what is going on in this individual as all of this is cycling. And was that Krebs?
That was Krebs. That was going up and down.
Krebs.
Yeah.
The other question is more, I do pain.
And in fact, apropos to that very quickly, that in animal models, again, it's harder to do in humans, but in animal models, if one gives drug continuously and not in a pulsatile manner, it looks like the drug is less addicting. It produces less downstream behavioral abnormalities in the animal than seeking the drug volitionally. So there does seem to be something to that pulsatile effect that is messing with the brain reward circuitry.
Yeah, the other question is, I do pain research, and one of the things that Frank Parecka has been pushing lately is that relief from pain, of course, is rewarding.
Yeah.
And you get a dopamine release in nucleus accumbens. But I don't know if people who become addicted to pain relief, they sort of like pain relief, but do you think those are totally different mechanisms happening in accumbens?
Boy, I don't know. I mean, that's an interesting point I hadn't thought of, Alan. It's clear that when I was in school, medical school, I was taught that you don't have to worry about giving an opioid to a patient in pain because a patient in pain will not become addicted to opioids. Wrong. So in some ways, anti-pain effects of opioids are much better understood now in the context of behavioral consequences for addiction. Opioids seem to be addicting regardless of whether they are alleviating pain, physical pain, or in a person who is not undergoing physical pain. Some people hypothesize maybe there's mental anguish that opioids are alleviating to produce a similar effect. Hi, Rob.
I've got a question trying to generalize a little bit. I'm just thinking about delta phos B and the histone variant. But if you have a conditional knockout of these, are there any general lessons you can learn from inactivating it? And is the global lethal?
No. So we have both. If you knock out delta phos B conditionally in an adult animal, D1 medium spiny neurons solely, the animals are fine. They seem, I hate to use the words depressed for a mouse, but they seem emotionally impaired and less rewarded by natural rewards, not only drugs. And so that just gives you one example.
Yeah, thank you for the great talk. This might be a naive question, but is there any benefit of this scarring mechanism you described? Is it like super useful to, I don't know, come back to very past experiences easily to learn certain things? Or I don't know. What would be the benefit of having, or is this just a purely artifact that?
Yeah, no, the way I'd answer that question is that I think the notion of chromatin scarring, which, basically some long-lasting changes in chromatin in response to a behavioral stimulus, is probably an evolutionarily conserved function mediating long-term adaptation in the brain, and that occurred to mediate positive adaptation. And it just happens to be that drugs of abuse are utilizing that mechanism and inducing a behavioral abnormality. But there is some evidence that similar types of mechanisms might come to play for long-term memory, for example. That the way a cell retains its ability to, so-called remember something is mediated through some of the same parallel mechanisms.
Eric, that was great. Thank you. Thinking about neurology and psychiatry and drugs and repurposing drugs, epilepsies are interesting and very complex disease, not as complex as use disorders, I think, but there are all these channel active agents, and I think they're effective because they affect many parts of the circuit problem.
Mm-hmm.
And that they are used much, much more in psychiatric disorders, having been developed for epilepsies, is so interesting to me. Do any of those drugs help with substance use disorders?
I am not the best person to answer that question. I'd ask folks in the audience. I mean, to my knowledge, the ability to use some of the anticonvulsants like topiramate or others to treat substance abuse disorders is not dramatic. In large studies, the effects are not so obvious, but some patients do report improvement. So there may be an effect there that is targeting a certain subset of people. Yes.
Thank you for this incredible work. I'm very curious about what you believe may drive the heterogeneity in the addiction index responses and the gene expression, given the inbred congenic mice, but such a heterogeneous response, number one. And number two, can you comment on any sex differences?
Yeah, sure. Absolutely. I could do both if I remember both. So the first question about why a genetically inbred group of mice would show so broadly different responses to a stimulus like cocaine is really interesting. The same is seen for any other type of stimulus. So if you take a C57 group of mice or any other inbred strain and characterize them with respect to cocaine or another drug of abuse, stress, foot shock, whatever it is, fear conditioning, one sees a broad range of responses. So that raises the question is what is mediating that diversity? And no one really knows the answer to it. The idea is that it represents stochastic random changes during development and the creation of the brain circuitry. That even on a fixed genome, even in the animal, in the experimental animal condition, the fixed environment, right? Because all the animals are pretty much raised together. You still get this diversity. So I personally favor that a lot of random events are occurring to specify brain circuits, and that is driving the diversity. It's not related to things like birth order in a litter of pups, because people have looked at that. It's not related toward the robustness of a pup when it's born in terms of size versus its litter mates and so on. So it's something different than that. In terms of sex differences, broad sex differences have been seen in humans and in rodents. In both, in general, the feeling is that females may tend to get addicted to a greater extent in a similar type of access condition. A lot that is in humans overlaid on culture, where certain drugs in olden days were less available to females. Now that has changed, and one is seeing this play out, I think, across pretty much all drugs, that females do tend to be more addictive under certain conditions. If one did when one does a transcriptional profile of various brain regions, Dina Walker in my lab has published on this, one sees broadly different transcriptional responses to the same drug exposure in females versus males, and within females, very large differences within the estrous cycle. So females tend to vary as much within the estrous cycle as the female-male difference at baseline. And again, trying to understand the basis of that is work that is just beginning to occur. Yes, thank you so much. Thank you.
These just get better and better. We'll see you here next year for the 8th annual Barondes lecture. Eric, thank you so much. Everyone, thanks for coming. Thanks for being so engaged. It's wonderful to see you. I think Eric has a few minutes to hang out down here in front if you didn't get a chance to have a question answered. Wow.